everybody. Thanks for joining this online seminar. My name is Steeve Boulant. I'm from the University of Florida. And today I'm going to talk about the impact of the hypoxic gut microenvironment in regulating host pathogen interaction. So first of all, I would like to acknowledge the two students that are participating in this work. So that work was actually initiated by Stephanie Munchau when the lab was still in Heidelberg, Germany. And we recently moved in the University of Florida, and the project now is continued by Sorin Jacob here on the right. So the intestinal epithelium is a complex interface. It is composed of multiple cell types organized as a monolayer. And you can see here the different cell types like the stem cell, the goblet cells, enterocyte, enteroendocrine cells, and they are organized in this crypt villi structure that you can see here on the left. And the cells, the crypt region, actually contain all the stem cells, and these cells are going to proliferate and differentiate and move along this crypt villi structure as they differentiate to form this nice epithelium. They are the primary barrier separating us from the outside environment. They are in constant contact with the ever-present commensal flora, and they need to tolerate the presence of the commensal flora while being fully responsive to pathogens. So to do so, the cells have developed many mechanisms to regulate how they interact with bacteria or enteric pathogen like viruses. And we know that this regulation of this finely tuned balance between immunotolerance and immunoresponsiveness can lead to chronic inflammation, inflammatory bowel disease, even cancer development. In the lumen of the gut, as you all know, there is a huge load, trillions of bacteria referred as the commensal microbiota. And they are very important for breakdown of dietary nutrient and also, for example, regulation of immune response. But with respect to our presentation today in hypoxia, so the oxygen environment in the gut, the microbiota is actually critical to establish and maintain this low oxygen environment that we have in the gut. The anaerobic microbes actually produce metabolite like butyrate that is actually consumed by our intestinal epithelium cells, and this consumption actually leads to oxygen consumption and basically decrease of oxygen level in the lumen of the gut. And for example, we know that disruption of the commensal microbiota, for example, when you take antibiotics, lead to a depletion of this oxygen hypoxic environment in the gut. And if you actually look at newborn babies that do not have an established microbiota, there's actually a higher oxygen concentration in the gut. If you actually now look at the tissue itself, this crypt villi structure, which I've mentioned at the beginning, the lumen of the gut, which is located at the tip of the villi, is, as I said, anoxic or do not have oxygen, do not contain oxygen. However, the crypt regions are highly vascularized, and it actually creates a normoxic environment of oxygen in the subepithelium. So if you actually look at this crypt villi structure, you can actually show and see that there is a gradient of oxygen that goes from the bottom of the crypt to the tip of the villi, placing the different cells, so the different epithelial cells, in different concentration of oxygen. Cells respond to low oxygen concentration by many mechanisms. The most important one and the most described one is through this hypoxia-induced factor 1, If1, which is composed of two subunits, the beta and the alpha subunit. When cells are in normoxic condition or normoxia, the proline hydroxylase, PHD, is going to take the oxygen in the medium, hydroxylate If1 alpha, and this hydroxylated form of If1 alpha can be recognized by the VHL protein, get ubiquitinated, and then degraded by the proteasome pathway. On the contrary, when cells now are in hypoxic condition, This hydroxylation through PHD cannot happen, and HIF1 alpha will be now stabilized, dimerized with HIF1 beta, and will act as a transcription factor and regulate transcription of about 100 different genes, which all have this hypoxia responsive element in their promoter region. Hypoxia is very critical for the gut. Not only it's important for the survival of the microbiota, so microbiota create hypoxia, and hypoxia makes the microbiota more happy, if I can say it this way. But our intestinal epithelium cells are actually benefiting from hypoxia too. For example, these intestinal epithelium cells made this factor called the threefold factor, which is actually upregulated in a HIF1 dependent manner. And this threefold factor is very important for regulating cell proliferation and barrier function in the gut. And that's very important in disease like inflammatory bowel disease. Because if we also have mouse, which for an example are knockout HIF1 alpha, they actually are not able to repair the damage that we can induce in an acute colitis model. So response to hypoxia by intestinal epithelium cell is very beneficial for the host when we are looking at the integrity of the barrier function of the gut. But what about hypoxia and how it regulates our host-pathogen interaction? Does hypoxia impairs how we sense and combat enteric pathogen? That's the question we are very interested in. And here I'm going to focus on the intrinsic innate immune response, which is generated by intestinal epithelium cells, which are always here marked as IECs. Every cell in our body can sense pathogen by pathogen recognition receptor. And this leads to a signal transduction cascade that leads to the production of cytokine. Among many cytokines, the one we are very interested in is the type 1 and the type 3 interferon. And these cytokines are secreted in the cell supernatant, in the environment, and they can act in an autocrine and paracrine manner to induce signaling through cytokine-specific receptor, so in our case, the interferon receptor, to induce interferon-stimulated gene. And this interferon-stimulated gene is what confers cell an antiviral state. There is three kinds of interferon, and we mostly focus on the type 1, the 2 interferon, the type 1 and the type 3 interferon. Both of these interferon have different receptor, the IFNAR1 and IFNAR2 for the type 1 interferon, and the interferon lambda receptor 1 and IL-10R2 receptor for the type 3 interferon. The type 1 and the type 3 interferon, the type 1 interferon receptor is ubiquity expressed in all cells, while the type 3 interferon is expressed mainly in epithelial cells and in a subset of immune cell. And this is why we are very interested about it when we are working on the gut, because this is the most important interferon that we have to protect us against viruses. Originally, people believed that both type 1 and type 3 interferon were kind of redundant, but over the years, many groups, including ours, have shown that the type 3 interferon is really specific to protect mucosal surfaces against enteric viruses. First of all, at mucosal surfaces like the gut, the cells that respond to type 1 interferon are mostly the immune cells, while the intestinal epithelium cells respond much less, while for the type 3 interferon, the epithelium cell responds very much to them, to all the interferon, to type 3 interferon, where the immune cell actually respond much less. And this actually leads to this model now that the type 1 interferon is important to protect against systemic spread of viruses, while the type 3 interferon is really important to protect the intestinal epithelium cells themselves. And this is not only true in a murine model, but also in a human model, where we can show that the type 3 interferon are really key in these cells. One last slide of introduction on the virus we are going to use today is Rotavirus. I think you all know about Rotavirus. It has been identified as a cause of diarrhea in 1973. And before, in the pre-vaccine area, it was the most common cause of gastroenteritis in children. Now, in developed countries, the vaccine is very efficient and rotavirus is not so much of a problem. However, it remains a huge health issue in developing countries where the vaccine is not readily available or actually not efficient. Like there is actually a region in Africa where the vaccine doesn't work very well and doesn't protect children against these viruses. And why do we care about rotaviruses? It's because it, as you can imagine, it's inducing gastroenteritis, and so it's affecting the gut. But if you actually look at the localization of rotavirus infection in this crypt villi structure, you can appreciate, so here you have in red cells which are infected by rotavirus, that the rotavirus infection is localized at the tip of the villi. And if we go back to this crypt villi structure where I mentioned that we have this gradient of oxygen where the tip of the villi is hypoxic, we found that very interesting that the region which actually have the less or the least oxygen are the ones which are infected by rotavirus. When people tried to study why is rotavirus infecting the tip of the villi, they actually use knockout animal system where the interferon lambda receptor, so the receptor for the type 3 interferon was knocked out. And what they could show is that the infection is now moving down the villi structure, demonstrating that this region here, circled in red, is actually protected by interferon lambda. But what about the tip? Why is it not protected by interferon lambda? Maybe hypoxia can impair the type 3 interferon-dependent protection of ISEs against rotavirus infection. And that's what I'm going to talk about today. And basically to put in the context of the gut, what I try to show you in the introduction is that in the gut, we have a commensal microbiota that is actually feeding our intestinal epithelium cells metabolite, like butyrate, that create or that induce our intestinal epithelium cells to consume oxygen and create hypoxia. It was shown in a completely different study that butyrate is actually able to decrease interferon production in cells. So what we are trying to address now is maybe this inhibition of immune response is due to the hypoxic environment which has been created through the consumption of butyrate. And if you have to remember just a couple of statements from this presentation, I would like you to remember that the low oxygen microenvironment in the gut impact the host pathogen interaction, rendering the host more susceptible to virus infection. I will show you that hypoxia regulates kinases and phosphatases that regulate signal transduction leading to interferon production. I will show you that hypoxia impairs production of interferon and that hypoxia promotes virus replication and spread. So when we started this work, we actually used not rotavirus, but we used another virus called mammalian reovirus, which is very similar to rotavirus, same family of viruses, but it's a non-pathogenic version of it. We infected cells that were grown under which were grown under normoxia or hypoxia. So throughout the talk, you will always see this graph in red and blue. Red means normoxia and blue means hypoxia. So we infected our cell with mammalian reovirus or MRV, and we actually first controlled that our cells were equally getting infected by this virus under the two oxygen condition. And as you can see here, as monitored by looking at the amount of virus genome using RT-PCR or monitoring the number of infected cells by immunofluorescence, we have the same amount of infected cells under hypoxia and normoxia about 16 hours post-infection. But if you now look at cells and monitor the immune response, we found something very interesting. We monitored the amount of type 1 and type 3 interferon that were made at the RNA level, so using RT-PCR. And you can see that between normoxia and hypoxia, there is a decrease in the production of type 1 and type 3 interferon when there is no oxygen in the medium. And this was not only true at the transcript level, but also true at the protein level, as we could identify this when looking at the amount of interferon that is secreted into the cell, by the cell into the medium. And as a consequence, we can also show that the downstream genes, which are regulated by interferon, the interferon-stimulated genes, are also decreased under hypoxia compared to normoxia. So that suggests that hypoxia suppresses the intrinsic innate immune response of cells, of intestinal epithelial cells, upon viral infection. We then went to the rotavirus experiments and basically repeated the same setup, and could actually show and the exact same finding that when we infect cell with rotavirus and monitor the amount of interferon that the cell produce, we do see a decrease between normoxia and hypoxia at the transcript, at the protein level, and this is also impacting the amount of interferon-stimulated gene that the cells are doing. So, similar to MRV, hypoxia decreases the interferon response following rotavirus infection. If we now follow how the rotavirus is able to infect cells, so here we are having a problem, and the movie doesn't seem to play, but I can show you the quantification in the next slide. Sorry, it's not moving. Here we go. So you basically have cells that we are infecting with rotavirus. So all the green cells that you see are infected cells. It's a rotavirus that encode a GFP fluorescent protein. And we basically follow live under normoxic and hypoxic condition infection and quantify the number of infection over time. So we're basically looking at spread of rotavirus in these cultures. And you can clearly appreciate here that under hypoxia, we have much more spread. So we have much more cells that gets infected over time. This is a 48 hours infection time course, and you can see it here in the quantification on your right. So hypoxia favor rotavirus spread in intestinal epithelium cells. And if we actually now look at the amount of de novo infectious virus particles that these cells produce, so here you have a plaque assay of rotavirus, and you can appreciate here that in the 10 to the minus 4 dilution or 10 to the minus 3, you have much more plaque in hypoxia at 48 hours post-infection compared to normoxia. So suggesting that we are really impacting the number of viruses that are produced during infection in hypoxic condition. And this is the quantification of the amount of virus, of infectious virus produced, so both released in the medium, so in the supernatant, or also associated with the cells, or remaining in the cells. And you can clearly see that hypoxia favor de novo infectious virus production and secretion. So because we found that hypoxia inhibits the immune response following MRV and rotavirus, we first wanted to validate that our findings were not virus specific, that were rotavirus specific. So we basically tested a completely different virus, Antivirus, which is used in the community a lot to study interferon response. And you can see the same phenotype here, that under hypoxia, we have less immune response. This was also true when we actually look and use the agonist poly-IC, which is the double-stranded RNA analog, which activate the Reg-like receptor pathway, or when we use the CGUMP to actually stimulate the STING pathway. So if we actually use completely different stimuli to activate different pathogen recognition receptor, we have the same phenotype that under hypoxia, we have less immune response. So it seems to be conserved between the different pathogen recognition receptor. The next step for us was to now validate that these findings were not cell type specific, because we were using intestinal epithelium cells derived from a colon carcinoma derived cell line called T84 cells. We thought that maybe because the cells are immortalized, we have a problem in signaling, so let's control that in human primary cells. And to do this, we actually use human organoid culture model. And the way we prepare our organoid is by isolating the intestinal containing stem cell crypt in the biopsies, and culture the stem cell in very specific culture condition to grow what we refer and what the community refer as mini-gut organoid. The advantage of this organoid culture is that they are primary, non-transformed human intestinal epithelium cells, and that they recapitulate the cellular complexity of the intestinal epithelium. So they basically contain the same cell type, like enterocyte, goblet cell, Paneth cells, that you actually see normally in the gut. So we took our organoid, and first of all, I can show you how they look like on microscopy. I can show you that we detect our different cell type. We have enterocyte, we have goblet set marked by mucin 2, we have enteroendocrine cells, we also have tight junctions, so they also recapitulate the barrier function. If we take this organoid and now infect them with MRV or Sendai virus, or treat them with the agonist poly-IC or CGAMP to activate the reglac receptor pathway or the STING pathway respectively, you can appreciate here that in all stimuli, we have less immune response under hypoxia compared to normoxia. So globally, even in primary epithelial cells, hypoxia negatively impacts the intrinsic immune response generated by epithelial cells. But why do we have this impact? So to find out where was the bottleneck under hypoxia, we decided to dissect the signaling pathway of interferon. So first of all, we focused on the paracrine signaling loop. We looked at whether signaling downstream the interferon receptor is impacted by hypoxia. To do this, we treated cells with interferon, either type 1, interferon beta, or the type 3, interferon lambda, and treated the cell with interferon under normoxia and hypoxia, and look whether we saw a difference. And here, interestingly, we don't see a difference between normoxia and hypoxia, telling us that hypoxia does not impact interferon-mediated expression of ISG, so it does not impact the signaling downstream the receptor. But maybe hypoxia, so it suggests that hypoxia is impacting something in terms of sensing of the virus or signaling downstream the virus or the pathogen. So we first focus on the different pathogen recognition receptor that we have in our cell, like the reglac receptor pathway with the RIGI MD5 or the different toll-like receptors, and monitor the expression under hypoxia and normoxia. So interestingly, for many of them, we didn't see any difference in expression between normoxia and hypoxia, or if we actually see a difference, we see that the pathogen recognition receptor are actually even more expressed under hypoxic condition. But that's kind of contradictory because I showed you that under hypoxia, we have less immune response. So we must have a negative feedback loop downstream this pathogen recognition receptor. We basically dissected this and first look at IRF3, which is a transcription factor that will be responsible for the transcription of the type 1 and the type 3 interferon. So when it gets phosphorylated, it goes into the nucleus, it binds the promoter region of interferon and transcribes this interferon. So what you can see here is, excuse me, a quantification of the level of phospho IRF3 or activated IRF3 following poly-IC treatment. And you can see that under hypoxia, we always have less phospho IRF3. So we have less activation of the signaling cascade. And if you now move one step above by looking at that TBK1 molecule, which will be responsible for phosphorylating IRF3, you can actually see the same phenotype that over a time course under normoxia and hypoxia, in hypoxic sample, we always find less phospho-TBK1 compared to in normoxic condition. So, and this was also true when we did a rotavirus infection. So we affected the cells with rotavirus and placed them under normoxia or hypoxia, and you can clearly see here that, again, under hypoxia, you have less TBK1, phospho TBK1, and less phospho IRF3. So hypoxia downregulate early IRF3 and TBK1 signaling upstream of the interferon gene. But how does hypoxia regulate this activation of IRF3 and TBK1? So this was, and this is basically kind of the summary, like a mini summary. So we know that we now have hypoxia. Hypoxia is actually blocking TBK1 phosphorylation, which in turn leads to less interferon and more virus infection. How do we do that? How does hypoxia does that? So we kind of focus on how does TBK1 get regulated into a cell? There's actually not much known about the regulation of TBK1 phosphorylation in the cell. This is kind of an ongoing field. But we basically dig in the reactome and interactome of TBK1, and we were looking for a protein for which the activity might be regulated by hypoxia itself. And we actually found a protein called casein kinase 2, or CK2, which is a serine threonine kinase. It's a very important kinase in the cell that is expressed in most of our cells. Interestingly, it was known that CK2 can negatively regulate interferon response. And it was also shown that the activity of CK2 can be regulated by hypoxia. But whether or not these two can be linked was not addressed, and this is where we actually started to dig. So for this, we basically use an shRNA approach, so knock down the expression of CK2 and repeated our MRV infection. And as you can see here, in my wild-type condition or in my shRNA scramble control condition, when I'm under hypoxia, I have less immune response compared to normoxia. However, when I do this in CK2 knockdown cells, I now have no difference between normoxia and hypoxia. And if you actually dissect the pathway and now look at TBK1 itself, at how it's getting activated. So first, in mock infected cells, when you make a knockdown of CK2, you can see that we have more phospho-TBK1. So proving that CK2 normally acts as a negative regulator of TBK1 activation. But if you do now this under poly-IC infection and you compare normoxia and hypoxia, so first of all, in wild type, just to remind you, in wild type cell, when I treat the cell with poly-IC, I see less phospho-TBK1 under hypoxia compared to normoxia. If I do that now with my CK2 knockdown cell, I see actually no difference. So CK2 negatively regulates interferon-mediated response by inhibiting the phosphorylation of TBK1 and the downstream induction of interferon gene. But the problem here is CK2 is a kinase, so which means this kinase needs to either activate or inhibit, the kinase that is responsible for phosphorylating TBK1, or CK2 needs to activate a phosphatase that actually will remove the phospho group for TBK1 and deactivate this protein. So here again, we were hunting for a protein that might also be regulated by hypoxia. And just basically to make a long story short, we ended up working on this protein called PP2A, or protein phosphatase 2A. It's an heterotrimeric protein phosphatase that have a very broad specificity. It's made of a catalytic subunit and here C, and a scaffolding subunit in yellow, and the orange one is the regulatory subunit. And there is many different kinds of regulatory subunit. The interesting fact about PP2A and regulation of immune response is that PP2A was shown to regulate TBK1 and IRF3 signaling, and it was also known to be recruited by host factor to shut down the IRF3 dependent type 1 interferon signaling. And most importantly, the activity of PP2A, or the level of PP2A expression, is varying in hypoxic conditions. So maybe there was a link there. So we went and address whether PP2A is involved of that. So we use a first strategy using a drug, so an inhibitor of PP2A, ocadaic acid. That's a very potent drug used in the field. And if you actually now use ocadaic acid, again, while in DMSO control sample, you can see that hypoxia blocked the immune response. In the ocadaic treated sample, you can see that you are actually now losing this inhibition. So really suggesting that PP2A is responsible for that hypoxic phenotype. We then perform a knockout of PP2A. So this is work ongoing. So we have only the polygonal knockout clone, so that's why you still see a little bit of PP2A left. So we knock out the catalytic subunit, the most important subunit. And you can see here that upon knockout of PP2A, we lose this hypoxia-mediated inhibition of immune response. So PP2A plays a key role in hypoxia-mediated inhibition of type 3 interferon. How does it do this? This is ongoing work, so I will not show you a final piece of data, but kind of want to give you a teaser for next presentation. We're basically looking at the expression level of the different subunits of PP2A under normoxia and hypoxia. And what you can see here is that the different regulatory subunits are differently regulated by hypoxia. So we have the 2A and the 5E, which are very differently expressed. And if you look in the one which are less expressed, you can actually see other subunits that may be a candidate. And currently we are focusing on the 5 subunit because it's highly expressed in the gut. So it seems to be kind of a gut-specific regulatory subunit. So to conclude, what I show you in this presentation is that hypoxia inhibits the intrinsic innate immune response generated by intestinal epithelium cells upon viral infection. So there is less interferon production. Hypoxia promotes virus infection and spread. The hypoxia-mediated inhibition of immune response is CK2 and PP2A dependent, and hypoxia negatively impacts TBK1 and IRF3 phosphorylation and the subsequent production of interferon. I apologize for that, it should actually be written interferon here. And if you look at it here, if I put this into a schematic, normally when you have virus infection in intestinal epithelium cells, you are sensed by the pathogen recognition receptor, activate phospho-TBK1, RF3, and then you make interferon, and that's basically fight the virus. However, under hypoxia, we have a CK2-PP2A pathway that will down-regulate this activation and promote virus infection. If I put this in the context of the gut, and now look back at the level of if I put this in the context of the crypt villi structure and look back at where are my hypoxic and normoxic region in my crypt villi structure, this is what we would like to speculate. If you actually are in the tip of the villi region, you are under low oxygen condition. That low oxygen condition is actually increasing the barrier function of the gut. This is work that has been done by others. We also have done some work on this. But what I just show you is we are decreasing interferon and ISG production. So we are making these cells in this hypoxic region more immunotolerant. And that makes sense because the tip of the villi is the part of the crypt villi structure which is the most exposed potentially to the microbial content of the gut. It's the closest to the commensal microbiota. On the contrary, when you look at the normoxic region of the gut, so here in the bottom, the crypt region, which contain the same cell, are under normoxia. So they have a stronger interferon response and stronger ISG production. So they are more immunocompetent. And that's very important because you want to protect this crypt region because having an infected crypt region will be catastrophic to repair the gut. So we really think that the hypoxic condition in the gut is a direct player in regulating this tolerance and protection against the commensal microbiota or against potential pathogen. And it's critical for maintaining homeostasis and creating a tolerizing environment. So I would like to to acknowledge my entire research group, especially Sorin and Stephanie that actually are working on this project, but also the rest of the team. And I would like to highlight that we are we are a husband-wife lab. I'm running a lab with my wife, Megan Stanifer, and we're really doing this work together. So thank you very much for your attention, and please don't hesitate to contact me if you have any question. I will try my best to answer your question.